In the basement levels of Uppsala University sits one of the oldest scientific institutions in Sweden, and for more than a century, it held something the public record barely acknowledged: a collection of catalogued human skulls, gathered not for medicine, but for measurement. The university's own ledgers once listed 57 skulls and 6 full skeletons believed to be Sami. Among them were 2 skulls dug out of a 13th century churchyard at Ronala in 1915, pulled from consecrated ground not to be honored, but to be measured against a theory. That theory belonged to the era of race science, and Sweden built entire institutions around it. Now, more than 100 years later, modern DNA sequencing of far older Sami-linked remains has quietly undercut the very premise those skulls were collected to prove. The population once forcibly measured to demonstrate its foreignness turns out, genetically, to be something else entirely. One of the last clear windows anywhere in Europe into how Siberian ancestry actually entered the continent. This is a real historical event, documented in the genome, not a slur invented to justify a museum drawer. What did the DNA actually find buried in those bones? And why did it take Sweden nearly 100 years to admit what those skulls in Uppsala were used for in the first place? Hit subscribe, because this one has bones, borders, and a paper trail, and every piece of it is real. The Sami have long been described, in amateur genealogy forums and outdated textbooks alike, as Europe's Asian people, a label that traces back to Cold War-era anthropology, and, before that, to 19th century race science itself. Whole genome analysis says otherwise. Multiple population genetic studies describe Sami and Finnish populations as phenotypically and genetically typical Europeans. The real finding is not a broad claim of Asian ancestry, it is something far more specific. A datable admixture event, roughly 3,500 to 4,000 years old, layered onto a much older European hunter-gatherer base. 19th century craniometrists used skull shape to argue the Sami were racially separate from the rest of Europe. 21st century genome sequencing shows the reverse. Deep European roots with one distinct, later genetic layer sitting on top. That layer has a name. It is Siberian-derived autosomal ancestry, not East Asian ancestry in the broad sense race scientists once assumed. That distinction never occurred to the men measuring skulls in Uppsala. Modern genetics insists on it. This matters for everything that follows, because the same misreading that got the science wrong is the misreading that justified taking skulls from a churchyard in the first place. The Sami register as more genetically distinct, more of an outlier, than almost any other population in Europe. But that outlier signal points toward a real Bronze Age migration event, not the racial otherness 20th century scientists assumed it proved. To see where that migration actually left its mark, the story moves north, to the Kola Peninsula, where the theoretical becomes physical. The paper is Lamnidis, Majander, and colleagues, published in Nature Communications in 2018, and it delivered the first ancient DNA ever recovered from mainland Finland. The team worked 2 sites. The first is Bolshoi Oleni Ostrov, on the Kola Peninsula in Russia, a burial roughly 3,500 to 4,000 years old. The second is Levanluhta, a water burial in South Ostrobothnia, Finland, around 1,500 years old, where the dead were interred directly in a lake, preserving bone in a way that Finland's acidic soil almost never allows. The research team combined the Max Planck Institute for the Science of Human History with the University of Helsinki, with Tisius Lamnidis and Kerttu Majander serving as co-first authors. In total, the study drew on 11 ancient individuals across both sites, a sample small enough to fit in a single case file, yet large enough to rewrite the region's genetic timeline. Levanluhta was not a random choice. Researchers selected it specifically because it stands as one of the oldest sites in Finland with well-preserved human remains, a rarity given how thoroughly the country's soil chemistry destroys bone elsewhere. 2 burial sites, separated by more than 2,000 kilometers and over 2,000 years, were about to be connected by something neither team expected to find in that quantity or that clearly. What surfaced in the laboratory did not match what anyone studying Iron Age Finland thought they already knew, and it would take the rest of the analysis to explain why. The individuals from the Kola Peninsula, dated to roughly 4,000 years ago, carried the earliest confirmed Siberian genetic ancestry found anywhere in Fennoscandia. That same ancestry resurfaces later in the population at Levanluhta, roughly 1,500 years old, hundreds of kilometers to the south. Central to the finding is the earliest known regional appearance of Y chromosome haplogroup N1C. a lineage entirely absent from every published Mesolithic genome recovered from Karelia and the Baltics. N1C is common today among Uralic-speaking populations broadly, which ties the genetic record directly to the linguistic history behind both the Sami and Finnish languages. Laid out as a timeline, the study shows Siberian ancestry reaching Kola by around 4,000 years ago, then spreading south into mainland Finland by the Bronze Age or Iron Age. The researchers frame this as evidence of a strong genetic connection between ancient Finnish and ancient Siberian populations, most plausibly explained by shared subsistence strategies and long-distance mobility across the north, not by conquest or invasion. The most surprising element of the paper was not the ancestry's Siberian origin. It was the location where the people carrying that ancestry turn out to have lived, far from where anyone expected to find them, and far from where their descendants live today. The individuals buried at Levenluhta turn out to be genetically closer to today's Sami than to today's Finns, despite the site sitting in South Ostrobothnia, well south of any Sami territory recognized today. That result confirms a suspicion linguists and place name researchers had already raised, based on Sami-derived toponyms still scattered across southern and central Finland. The implication is direct. A Sami-like population once occupied a far larger stretch of Finland during the Iron Age than at any point since. In the centuries that followed, agricultural expansion and Finnish in-migration appear to have displaced or absorbed that southern population, rather than blending evenly with it. This lines up with an earlier mitochondrial DNA study by Der Sarkisian and colleagues, which documented genetic discontinuity between prehistoric Northwest Russian populations and their present-day descendants, consistent with episodes of population replacement rather than smooth, unbroken lineage. The southern Sami genetic and linguistic footprint effectively vanished from the historical record until ancient DNA brought it back into view. The usual telling of Sami territorial shrinkage frames it as a story confined to the last few centuries of colonization, but the genetic evidence pushes that contraction back more than 1,000 years earlier than most accounts allow for. That contraction, though, is only half of what the DNA shows. The rest of the story is written into the genetic signature of the population that never left at all. 60%. That's the frequency at which Y chromosome haplogroup N1C shows up in Sami populations, one of the highest concentrations recorded for this lineage anywhere in Europe. For comparison, that same lineage reaches similarly extreme frequencies far to the east among Siberian and Uralic populations, 58 to 94% among the Nenasan, 82 to 95% among the Yakuts, 75 to 93% among the Nenets. A study by Sykes and Achille, examining Y chromosome markers from nearly 3,000 individuals across 32 populations, identified just 2 major founding male lineages behind the entire modern Sami paternal gene pool. 2 founding lineages, sitting behind a population of roughly 80,000 people today, is a textbook signature of a founder effect or population bottleneck. That bottleneck most likely reflects a small original group size, geographic isolation, and centuries of endogamy, not the racial purity older theories claimed. Researchers behind the study explicitly described the Sami as extreme genetic outliers among European populations, based on this Y chromosome and mitochondrial DNA structure. That phrase, extreme genetic outlier, was a technical description of population genetics. Race scientists later stripped it of that context and weaponized it as evidence of foreignness. The maternal side of this story runs even deeper, and considerably older than anything found on the paternal line. Mitochondrial haplogroup U5B1B1 exceeds 40% frequency in some Sami subgroups, a concentration far above any other population in Europe. U5 as a broader lineage ranks among Europe's oldest maternal haplogroups, documented in human remains more than 30,000 years old elsewhere on the continent. The other Sami maternal hallmark, haplogroup V, traces back to a post-glacial population expansion out of the Franco-Cantabrian refuge, in what is now northern Spain and southwestern France. Tambets and colleagues, in a 2004 study, calculated the divergence time for Sami haplogroup V at roughly 7,600 years before present. The same study dated the divergence of U5B1B1 at roughly 5,500 years ago within Sami populations alone, and roughly 6,600 years ago when shared between Sami and Finnish populations. 2 deep, ancient maternal lineages sit alongside one much younger paternal lineage, N1C, which arrived only 3,500 to 4,000 years ago. The male and female ancestral histories of the Sami simply do not line up on the same timeline. A north-south divide inside Sweden itself makes that mismatch even messier to untangle. Northern Swedish, Norwegian, and Finnish Sami populations are dominated by haplogroups V and U5b1b1. Southern Swedish Sami show a markedly different mitochondrial profile, one that sits closer to the general continental European population than to their northern counterparts. The Tambets study links this directly to occupation and lifestyle. Southern Swedish Sami, when stratified by livelihood, show the admixture pattern concentrated specifically among those in closer, longer contact with settled Swedish society. This complicates any simple genetic definition of who counts as Sami, a question carrying real legal weight today in reindeer herding rights disputes and Sami parliament enrollment decisions. It also means the DNA itself resists the same essentialist framing that 20th century race scientists tried to impose on it from the opposite direction, treating it as a fixed marker of racial otherness. The lesson cuts against both the old misreading and any tempting new one. Genetics here tracks history and contact, not a fixed racial category in either direction. None of this ancestry traces back to a single point of origin. It has an origin story that reaches back before pottery existed in the region at all. Before any Siberian-derived ancestry arrived, the region was home to the comb ceramic culture, active roughly from 4200 to 2000 BCE across northeastern Europe. Comb ceramic communities were hunter-gatherers who adopted pottery without ever adopting agriculture, producing large storage vessels holding 40 to 60 liters, decorated with comb-stamped and pit impressions. Their settlements were seasonal, built near rivers, lakes, and coastlines, with dwellings of around 30 square meters housing roughly 15 people at a time. Burial practice involved covering bodies in red ochre, and the archaeological record points to an egalitarian social structure, with no elite graves and no hierarchy visible in grave goods. Along the southern Scandinavian coast, a related tradition, the pitted ware culture, active from roughly 3500 to 2300 BCE, persisted north of the agricultural funnel-beaker culture, sharing ceramic-style roots with the comb ceramic groups further north and east. This is the population substrate that the Siberian ancestry layered onto starting around 4,000 years ago. It was not a blank slate. It was an already established hunter-gatherer world with its own deep roots, its own burial customs, its own material culture stretching back over 2,000 years before any new lineage arrived. Understanding that layered history is exactly what the race biologists working in Uppsala got backwards. In 1921, the Swedish parliament approved funding for what became the world's first state-sponsored institute dedicated to race biology. The Statens Institute for Rasbiologi opened its doors in Uppsala on the 1st of January 1922, housed in a building called Dakenhuset. Support for the founding proposal crossed the entire political spectrum, with both Hjalmar Branting, the Social Democratic Prime Minister, and Arvid Lindman, the Conservative Prime Minister, backing it. Its first director, Herman Lundborg, was a physician and psychiatrist whose central research obsession was the risk of what he called race mixing between Swedes, Finns, and Sami in the north of the country. Over its operational years, the institute conducted craniometric and biometric measurements on an estimated 100,000 people, drawn overwhelmingly from military conscripts and minority populations rather than any representative national sample. This was not a fringe operation. It inspired similar race science institutions abroad and operated with full state backing for over 3 decades. What that institute did with actual Sami bodies, both living and dead, is where the story turns uncomfortable. Return to that basement in Uppsala, now with the full weight of what it actually held. The university's Department of Anatomy accumulated Sami skeletal remains for decades, primarily skulls dug from Sami burial grounds and brought back for study. Old university catalogs list 57 skulls and 6 skeletons identified as Sami origin, stored alongside the remains of prisoners whose bodies were handed over to the university by the state, a practice that continued into the 1950s. 2 of those skulls, dug from a churchyard at Runala in 1915, are believed to belong to individuals who lived in the 13th century, remains taken not through accidental grave disturbance, but by design, specifically for measurement. Those remains were put to use in craniometric comparison, measuring skull shape and size in an attempt to prove supposed differences between what researchers called Nordic Swedes and the Sami population. In 2007, the Sami parliament formally demanded the return and reburial of all Sami remains still held in state and university collections, a demand grounded directly in international indigenous rights law. As of recent reporting, Uppsala University is still working to positively identify which remains in its holdings are actually Sami, because decades of poor cataloging mixed them in with unrelated collections. Measuring the dead was only the first phase of the program. The second phase targeted the living. In 1934, Sweden passed a law authorizing involuntary sterilization on eugenic, social, or medical grounds, built directly on the racial biology research the Uppsala Institute had spent over a decade producing. That law remained in force until 1976, 42 years, well past the point at which Nazi Germany's own eugenics programs had already been globally discredited by the war. A government-commissioned inquiry in the 1990s found that up to 63,000 people were sterilized under the program, roughly 90% of them women. Grounds for sterilization ranged from what officials called feeble-mindedness to poverty to being deemed of mixed blood, a category that directly implicated Sami people and other minority Swedes. Sweden was not alone in this. Comparable eugenic sterilization programs ran in Norway, affecting roughly 40,000 people, and in Denmark affecting roughly 6,000, over the same decades, all under social democratic governments. The scale of it only became public knowledge in 1997, after a series of investigative newspaper articles in Dagens Nyheter broke 4 decades of institutional silence. The program did not end quietly in the 1940s alongside Nazi Germany's collapse. It continued for another 3 decades, running well into an era most people now associate with modern, progressive Scandinavian welfare policy. The reckoning that eventually followed was considerably smaller than the harm that preceded it. After the 1990s parliamentary inquiry, Sweden's government agreed to compensate victims of the sterilization program. Of the tens of thousands affected, roughly 63,000 by the official estimate, the state ultimately compensated just 3,000 people. Then Minister for Social Affairs Margot Wallström publicly called the program nothing but barbaric in 1997, the first senior state acknowledgement of what had happened, arriving decades after the practice itself had ended. The Council of Europe's Parliamentary Assembly formally confirmed in a 2011 report that Sweden's eugenic sterilization laws created over 60,000 victims. Filmmaker Kjell Sundstedt discovered only as an adult that 4 of his own uncles had been forcibly sterilized under the program, a story his family had never discussed, out of shame at what had been done to them. The State Institute for Racial Biology itself was never shut down in disgrace. It was quietly renamed the Institute for Human Genetics in 1958 and absorbed into Uppsala University, where its successor department still operates today. Norway's version of this history did not stay buried nearly as long, but it followed a strikingly similar shape. Norway pursued its own decades-long assimilation campaign against the Sami, alongside the Kaiven, or Norwegian Finn, and Forest Finn minorities, a policy known as Fornorskningspolitiken, or Norwegianization. Boarding schools sat at the center of the policy, with Sami children removed from their communities and punished for speaking their own language. In 2018, Norway's Storting established a Truth and Reconciliation Commission, chaired by Dagfinn Høybråten, to formally investigate the policy's history and lasting effects. The Commission's final report, delivered on the 1st of June 2023, ran 700 pages and drew on interviews with more than 700 individuals. Its most frequently cited themes were language loss, discrimination, schooling, and fractured identity, harms that living Sami Norwegians still actively report today, not confined to history. In November 2024, the Norwegian Parliament issued a formal state apology to the Sami, Kjønven, and Norwegian Finn, and Forest Finn peoples, the first apology of its kind in the country's history. 2 countries, 2 commissions, 2 apologies decades apart, and the genetic research into the same population remains tangled up in the aftermath. Under the EU's GDPR framework, ethnic origin combined with genetic or biometric data is classified as sensitive personal data, meaning Sami genetic research today carries legal weight most population genetic studies simply don't. Researchers working in this field have documented their own explicit hesitation. A 2023 Frontiers paper on open science in Sami research describes scientists actively debating whether to publish Sami genetic data openly at all, given how that kind of data has been misused historically. A widely cited cautionary case from elsewhere in indigenous genomics involves DNA collected from the Havasupai tribe in Arizona in 1989 for diabetes research. later reused without consent for studies on schizophrenia, inbreeding, and migration history, a breach that prompted the tribe to disengage from biomedical research entirely. The practical result is that many geneticists studying Sami ancestry today keep a careful distance from the race biology history in their own papers, treating the two as separate stories, even though one directly enabled the other. Collective consent remains the unresolved problem at the center of all of it. Individual research participants can consent for themselves, but genetic findings inevitably say something about relatives and communities who never agreed to anything. This is precisely the gap the Human Genome Diversity Project ran into decades earlier, as indigenous rights organizations worldwide objected to being treated as objects of scientific curiosity rather than partners in the research. None of that caution erases the science. It just means the story has to be told carefully, which is exactly what the DNA itself demands. 6 layers make up this history, and laid end to end, they finally explain everything that came before. Layer 1 is a comb ceramic hunter-gatherer population, present in the region from at least 4200 BCE. Egalitarian, pottery-using, and non-agricultural. Layer 2 is a Siberian-derived genetic influx beginning at the Kola Peninsula roughly 4,000 years ago, carrying Y chromosome haplogroup N1C into Fennoscandia for the first time. Layer 3 is that same ancestry spreading south into mainland Finland by the Iron Age, reaching as far as Levenluhta, hundreds of kilometers from any Sami territory recognized today. Layer 4 is centuries of contraction, driven by agricultural expansion and later by state assimilation policy, compressing that footprint down to the roughly 80,000 Sami people spread across 4 countries in the present day. Layer 5 is the 20th century race science that misread that entire layered history as evidence of a fixed, separate, so-called Asian racial category, and then acted on that misreading through sterilization law and grave robbery. Layer 6 is 21st century ancient DNA correcting the record, even as the ethics of doing that research remain genuinely unresolved. Every layer of this history, the migration, the misreading, the harm, and the correction, is legible almost entirely because of DNA extracted from the dead, the same category of evidence race scientists once extracted for the opposite purpose. The living population carries all 6 of those layers at once, in ways modern medicine is only beginning to reckon with. The same founder effect and endogamy that produced the extreme N1C and U5B1B1 frequencies also concentrates rare recessive genetic conditions within the Sami population, a pattern documented in several regional health genetic studies. Because Sami genetic data is legally sensitive and historically fraught, Sami populations remain underrepresented in the large biobanks and reference panels that modern precision medicine increasingly relies on. That underrepresentation is not hypothetical. It means diagnostic tools calibrated on majority European reference data can perform less reliably for Sami patients specifically. Indigenous genomics researchers globally have flagged this exact tension, noting that the same populations most harmed by historical genetic exploitation are now the populations most excluded from genetics-based healthcare improvements. Sami community leaders and researchers have pushed for data sovereignty frameworks, meaning Sami-led control over how genetic data about Sami people is collected, stored, and used going forward. The debate remains unresolved in every country with a Sami population, precisely because none of the 4 states involved has built a genetics research framework that the Sami parliament itself endorses. The population at the center of all this research still lives across 4 national borders, on land that was never divided with their history in mind. Sapmi, the Sami homeland, stretches across northern Norway, Sweden, Finland, and Russia's Kola Peninsula, a region with no single government, no unified census, and population estimates ranging anywhere from 50,000 to 135,000. The most commonly cited figure sits at roughly 80,000 people, with Norway home to about half of them. 9 to 10 distinct Sami languages are spoken across that territory: Northern, Lul, Southern, Umi, Pite, Skolt, Inari, Kilden, and Ter, while Akala and Kemi Sami are now extinct. Only an estimated 2,600 to 3,000 people across the entire population still make their living herding reindeer, managing roughly 213,000 tame animals in Norway alone. Reindeer herding rights remain actively contested, with recent Norwegian Supreme Court rulings on wind farms built on traditional herding land drawing active Sami protest as recently as 2023. The population that Herman Lundborg's institute spent 13 years measuring for signs of racial difference is, genetically, one of the clearest surviving records of how ancient Siberian migration actually reached Europe. Every unreturned skull in that Uppsala collection still represents someone whose descendants are alive today to ask for them back. Uppsala University's project to positively identify its Sami remains is still ongoing, and the big pile of bones, in one researcher's own words, has resisted full sorting for over a century. The Levenluhta site itself hasn't been exhaustively sampled either. Only a fraction of its burials have been sequenced, meaning the southward extent of ancient Sami-related ancestry in Finland is still only partly mapped. No comparable large-scale ancient DNA study has yet been published for the Norwegian or Swedish mainland Sami population specifically, leaving major gaps alongside the well-studied Kola and Finnish material. The unresolved consent question, how to conduct genetics research on a population with this specific history without repeating its extraction, has no agreed answer among the researchers currently working on it. What is settled is that the Siberian ancestry is real, dated, and traceable. What remains unsettled is who gets to decide how that story is told, studied, and used going forward. The DNA has already answered where the Sami came from. It hasn't answered who gets to keep asking. Picture that drawer of skulls in Uppsala one more time. Some now identified, some still unmarked, sitting in a collection that has outlived the institute that built it. The core finding stands on its own, a genuine, dated migration out of Siberia, roughly 4,000 years old, shaped the modern Sami gene pool. That is real history, obscured for decades by a race science that got the meaning of that history badly wrong. One tension remains unresolved to the end. The same tools now correcting the historical record are the direct descendants of the tools once used to justify harming the people that record belongs to. What's left, at the close of it all, is not the controversy but the population itself. Roughly 80,000 people across 4 countries, 9 surviving languages, a shrinking number of reindeer herders, and a genetic signature still being read for the first time on its own terms. When the science and the history finally get told together, honestly, who should be the one telling it? That's the question Sapmi is still waiting on an answer to.